have discussed today by JJ that dapagliflozin has stormed the market. That is just possible because the price was reduced and the innovator like molecule came to the market. Similar thing is going to happen in insulins also, has already happened. These biosimilars of insulin are going to storm the market and get a lot of benefit to the poorer people, those who cannot afford original. And if you look at the insulin story from Joseph von Mering, who recognized that removing a pancreas can cause diabetes. Then Himsworth in 1936 recognized insulin resistance in type 1 diabetes. Benting and Best in 1921 they really discovered insulin, and with MacLeod and others, and David Godel he used E. coli for the recombinant production of insulin in 1978. So, if you look at the evolution of a therapeutic breakthrough of insulin, you can see from 1922, where a commercial thing was, the first was 1923, commercial insulin was available, until 1990 to 2000. You can see recombinant urine was insulin was in 1980. Then 90 to 2000 saw a lot of changes like insulin, detamir, degludec, all those things started coming. Insulin aspart and new things. And after the new generations are started coming now, like IDEG, IGLAR, and other Ultralente, and IDEC Lira, and inhaled insulin have also come. And many things are coming in the future also. And they will change the scenario of insulin use in this country. So, like coming are once a weekly insulin, oral insulin, liver preferential insulin, glucose dependent insulin, and beta cell replacement. These things are going to change the way we have been handling the insulin therapy. But here comes a good news: India's unique opportunity and important responsibility as the pharmacy of the world. We can see that a team of very dependable developers and promoters have now started producing the biosimilar of insulin aspart or Novorapid's biosimilar. What is a biosimilar? Providing the three A's of insulin like affordability, availability, and accessibility. Access to affordable insulin is critical. Insulin products like insulin glargine, Lispro, Aspart play an important role in the treatment of type 1 and type 2 diabetes. Biosimilar and interchangeable biosimilar products have potential to reduce healthcare cost, similar to how generic drugs have reduced cost in oral drugs. Biosimilars marketed in the US typically have launched with initial list prices 15% to 35% lower than comparative list prices of reference products. This is very important. So, what is a biosimilar? Biosimilar is similar nature to reference product in terms of quality, safety, and efficacy. Biological product is highly similar to reference product. No clinically meaningful differences between the two. Similar biotherapeutic products is a biotherapeutic product that is similar in terms of quality, safety, efficacy, and already licensed, and has been proved by MEA, USFDA, WHO, and DCGI. A similar biologic product is that which is similar in terms of quality, safety, and efficacy as per DCGI. So, based on comparability. So, requirement for a biosimilar is many. We can touch upon like primary structure, higher order structure, product variants, metabolic activity, immunogenic activity, and purity has to match shoulder to shoulder with the innovator. And if you look at biologics and biosimilars, you can see the manufacturing is almost same in manufacturing, immunogenicity, regulation indications, potential for patent licensing, potential for exclusive period, and potential for interchangeability is there, but licensing biosimilars do not have potential for patent licensing, and potential for exclusivity period is not there with biosimilars, whereas that is 12 years for biologics. Here is a name, very important now in the Indian diabetic scenario: insulin aspart injection, Insuquick, the power of choice, 100% made in India, the first biosimilar insulin aspart in India. 100% indigenous and robust technology, global quality in products, processes, and dossiers, global standard, clinically safe, and efficacy trials. This has passed all. The CGMP production facility is there, good manufacturing practice, strong intellectual property portfolio for value creation. Insuquick or insulin aspart is meeting all these criteria and preconditions. So, first biosimilar recombinant insulin Aspart was given approval in the year 2021 by DCGI. 2021 it has been approved, but recently it was launched. First biosimilar insulin aspart in India or Insuquick has a fingertip as if thumbnail impression like processes, establishes quality biosimilarity of its products by characterization with fingerprint like similarity using several advanced technologies recommended by global regulatory authority, like in primary structure, higher order structure, biological, purity, and impurity. Everywhere it is matching, and it is one of the best products now. If you look at the global standard of clinical safety and efficacy trial, GCP compliant and EMA compliant clinical studies, statistically significant and meaningful clinical trial sizes, the pharmacokinetics or PK, it has been compared with Novorapid in orange and Insuquick in blue. And you can see they match shoulder to shoulder everywhere, all as if the same molecule. And also clinical trials pharmacodynamics has been seen, and it has been compared with Novorapid to Insuquick, and you see the pharmacodynamic also is matching so concurrently with Novorapid. So this is a true replacement of Novorapid when needed. The first biosimilar insulin aspart in India, that is Insuquick, is strong IP portfolio for value creation. IP portfolio or patents at biogenomics, India, PCT, Europe, US, Malaysia, and Singapore. So, international intellectual property right and patent cooperation treaty is in place. Innovations covers super vectors for mammalian and microbial expressions, codon optimized gene sequences, protein folding technologies, novel purification and isolation technologies. So, what is the significance of evidence? Residual uncertainty, stepwise approach to generate quality of evidence, and additional clinical studies for establishing product safety and efficacy is still underway. So you see physiochemical and biological characterization is established, preclinical studies are established, clinical pharmacology PKPD study is established, and studies are still going on to prove that this is This is a safe insulin, effective insulin, but still some studies are going on. Stepwise approach for biosimilars development program is always there, like any extensive structural and functional quality determination, biosimilarity, animal toxicologic studies, phase 1 human clinical study like PKPD, phase 3 human clinical study like interim comparative safety and efficacy, and then only marketing authorization is available to market the product. Once these criteria are met and it is proved to be a safe and effective biosimilar, then only the permission is available. As we discussed earlier, structural characterization of insulin aspart, analysis of primary, secondary, tertiary, and quaternary structural attributes using orthogonal spectroscopic tools, and it came out to be successful. Structural analysis of Insuquick has been done in primary structure, secondary structure, quaternary structure, and tertiary structure. And this proves a peptide formed under non-reducing conditions and peptide formed under reducing conditions have been compared, and the molecule has proved to be one of the best till now. These are all little technical diagrams, not we'll discuss later, but it proves that spectra confirms high similarity between BGL ASP and reference product. So spectroscopy and other things have been done, and the reference product and this Insuquick molecule, they prove at par. NMR studies have been concluded like 1D, COSI, TOXI, NOISI, HSQC, spectrum acquired during NMR study for BGL aspart and reference, and the product are identical to each other in solution state at molecules at atom to atom three-dimensional structural levels. So this is comparable and strongly suggests structural similarity of primary, secondary, and tertiary structures. Physiochemical characterization has been already studied, like impurities have been worked out, and there is very little impurities in this. This is a very, very pure product. And also, physiochemical characterization of Insuquick has been studied vastly. Assay content and related impurity content and HMWP content are found to be in compliance with identified biosimilarity limits, which are more stringent than Indian pharmacopoeia specifications. In excipient analysis, phenol, M-cresol, pH, and zinc content are found to be highly similar, but the reference and Insuquick batches in stability studies, Insuquick is stable for 30 months at 2 to 88 degrees centigrade and is found to be highly similar. This is a great study, and this gives a lot of confidence to the clinical physicians to prescribe the product. So biological characterization of recombinant insulin aspart. So this is also there. Insulin bioassays have been proved. It measures affinity of insulin for insulin receptor, measure for phosphorylated insulin receptor, glucose uptake, lipogenesis, inhibition of lipolysis, and mitogenesis cell proliferation have been studied and estimates mitogenic potential in cells, and they have got all good result. In vitro biological characterization of recombinant insulin aspart from biogenomics and originator insulin aspart has been studied, and a paper by Misra et al. They suggest that the binding studies showed that the affinity of BGL aspart was highly comparable to novorapid, insulin receptor autophosphorylation, glucose uptake, and lipogenesis demonstrated high similarity to the RMP. The mitogenic assay for BGLAS did not show any proliferative effect and was comparable to the RMP. The in vivo bioidentity test showed that BGL aspart is highly similar to the innovator Novorapid. So conclusion of in vitro biological assays has been done and extensive biological characterization demonstrates a high degree of functional similarity between the biosimilar insulin aspart from biogenomics or InsuQuick and reference innovators product. These are all detailed studies. Preclinical animal toxicology studies comparatively established in extensive preclinical and animal studies was done. Then product development program took a long process comparatively in human phase 1 PKPD glucose clamp study to show biosimilarity of insulin aspart to reference product. Pharmacokinetics and pharmacodynamics was studied. Phase 3 clinical study design was very robust and it efficacy, safety and results were proved in a 24-week administration of Insuquick and reference product were comparable and equally effective in lowering HbA1c, FPG, PPG and type 2 diabetes in type 2 diabetes patients. So the study concluded that established similarity in between reference product and BGL aspart in reduction in immunogenicity and neutralizing antibody assay proved absence of neutralizing antibodies in positive clinical samples. So there have been so much publication of Misra et al. In endocrinology and investigations in 2023, which says comparative assessment of immunogenicity of recombinant insulin aspart from biogen originator Novorapid in adult patient with type 2 diabetes. Immunogenetic studies for Insuquick has been studied, and the endpoint was to compare immunogenicity of recombinant insulin aspart manufactured by BGL with a reference product by measuring antibodies at baseline, end of 12 weeks, and end of 24 weeks. And the baseline 3 months and 6 months was studied. Quick check for antibodies against insulin aspart. There was no. If key question, are these antibodies neutralizing in short? Do antibodies affect efficacy of drug? So that was found out, and during all those things it was found it was comparable, and incidence of detectable antibodies was found during visit 821. Patients show immunogenicity and immunogenic response showed by these patients was very low in terms of antibody titer. So serum samples analyzed for neutralizing antibody assay was studied. During visit 8 and 21, patients show immunogenicity and immunogenic response showed by these patients was very low in terms of antibody use. Average glucose consumption of patients who received reference product as treatment was in range of 80.34 to 83.48, whereas patients who received Insuquick aspart was in the range of 80.27 to 82.63, better result. So these are the two devices that is used. USB pen, straight USB pen. It is a reusable pen. You can put the cartridge of Insuquick and this can be used like any other pen and a reusable pen. And the disposable pen name is Insuquick VD pen, and this can be disposed and it contains the product Insuquick, and this is easy to use. Both the pens are very easy to use. So successful marketing authorization from DCGI, CDSCO as fast biosimilar insulin aspart in India has been obtained. Impact of Insuquick on diabetes management in India is it is more cost effective option, making diabetes management financially acceptable to a wider population, and reduce expenditure on diabetes care due to affordable insulin options. Access to effective and affordable insulin plays a key role in reducing complications associated with diabetes, enhancing the overall public health scenario in India. So, thank you, dear friends. Now we'll go to the panel discussion.